Okay, I think we're recording now. All right, so this is immediate brain plasticity after 1 hour of brain-computer interface. I'm sure most of you know what a brain-computer interface is, but just as a reminder, it's basically a, uh, a piece of technology that allows us to directly communicate between a human brain, or actually any brain really, even an animal brain, and a computer, so like a computer program. It can be either invasive or non-invasive, right? In this one, I believe this is a non-invasive EEG-based BCI. But you can also have invasive brain-computer interfaces where you like stick needles into someone's cortex, right? So they had these participants use a brain-computer interface for an hour, completely inexperienced, right? And before and after they underwent that hour of BCI usage, we basically, or the authors basically ran an fMRI over their brain, and what they found is that after just 1 hour, there were significant changes in the structure of the brain, and yeah, they basically found that depending on what type of BCI they used, right, like motor imagery BCIs versus VEPS BCIs, they found that the changes in brain plasticity are spatially specific to the respective neurofeedback, aka for the BCI that use motor imagery, they found increases in plasticity in motor correlated regions of the brain, whereas with the VEPS BCI, they found the changes were primarily correlated with like visual parts of the brain, like the occipital cortex. And yeah, so the spatial specificity promises tailored therapeutic interventions, right? So if we know that a certain part of the brain does something X, right, then we can develop a BCI that specifically targets that task or that function, and then we can stimulate what's it called, plasticity changes in that part of the brain. So yeah, any questions about any of these fundamentals or the basic overview before we jump into this? The basic idea of this paper is pretty straightforward, I think. It's more interesting in terms of the methods it uses, right, and like the specific type of changes they uncover.
I have a question.
Yeah, sure, go ahead.
Could you briefly go over what brain plasticity is?
Sure. So neuroplasticity or brain plasticity basically just means the ability for your brain to change, right? So if the process of learning itself depends heavily on neuroplasticity. When you're learning, the neurons in your brain reconfigure each other, right? They connect with new neurons, or the structure, the structure that connects neurons to one another changes. So brain plasticity or neuroplasticity is the ability for your neurons to adapt to new information. And here, I'm not like by fMRI, I'm pretty sure, since it's fMRI, they weren't looking at specific like individual connections between neurons, so they can't say like, oh, neuron A used to be connected to neuron B, but now it's connected to neuron C, so they can't say that. But they can look at these overarching patterns and see how the activations in neurons changed before and afterwards. That's implicitly due to the plasticity of the brain, right? You started out with the activations happening in pattern A, then you stimulate the brain with the BCI for 1 hour, just 1 hour, right? And because the brain is plastic, that region of the brain that's being stimulated by the BCI is going to change. And then after the hour is up, then when you look at the firing patterns of the brain, they're different. They now fire in like pattern B. Does that make sense?
Yeah, I got it. Thank you.
Okay, cool. Yeah, any other questions before we dive in? Cool. Okay, so right, this is a bit more in-depth background, right? A BCI allows humans to control computational devices. Do BCIs impact the brain? They show some spatially specific signs of brain plasticity, modulations of EEG rhythms by motor imagery, right? So they call that the MIBCI, or ERPs, or like, remember a few weeks ago we were talking about like ERPs versus evoked potentials. They're pretty similar and they're sometimes used synonymously, but they're technically different. But here I think they're using them basically synonymously. So ERPs, right? So one is visual and one is motor. And they show that, yeah, so then like actually measuring wise, they perform structural and functional MRI. They'll probably give more details later on about those methods. They found increased T1 weighted MR signal in the gray matter of the respective target brain regions. So this is probably the most jargony sentence here. But basically, I think they're saying that they found increases in this pattern, right, in the occipital regions, right. The occipital lobe is correlated with visual processing when they use this visual stimulus, but the sensory motor region when they use the motor imagery stimulus. So this is the pattern that they basically discovered is correlated with usage of this BCI. And they also show that there's increased functional connectivity for the motor imagery one. Yeah, exactly. So now we can basically, we know there's a way to use BCI to directly impact certain parts of the brain in terms of structure and function. And yeah, there's potential therapeutic applications of this. Cool. So yeah, here they're just going over what a BCI is, right? An important component is the neurofeedback, right? You give some information to the system, right, by like thinking, and then the system feeds information back to you into your brain somehow. So here they talk about the two types of approaches they use, right? The two types of BCIs, the motor imagery BCI and the ERP BCI. So for the ERP one, it's induced by observing and counting flashing letters on the screen, right? And that's obviously like a visual stimulus. They rapidly achieve good performance and it like plateaus. The sensory motor rhythms, on the other hand, is a more complex task. Yeah. So about 30% of the users for the motor imagery task are actually unable to gain control, it seems. So it's definitely a harder one. And motor imagery basically means that you are imagining yourself doing something. So in your brain, you basically imagine yourself moving your hand or like moving your leg. And then we can use a BCI or like these EEG signals to pick up on those signals in the brain and predict like what you want to do with your body, or like what you're thinking, what you're imagining, what you're imaging yourself doing. Yeah. So yeah, recent progress has improved this motor imagery BCI stuff, right? There's plenty of applications. Yeah, they talk about the therapeutic uses again. Yeah, so this is how they quantify their fMRI stuff, right? So there's like a ton of stuff over here. I was sort of talking about this earlier as well, right? Like how can we tell with an fMRI whether there's actually changes in brain plasticity or not? So they have like a ton of citations over here that sort of show that using an fMRI can actually reveal changes in brain plasticity for both motor and other types of tasks. Right, so then there's like some special type of fMRI that they use, like task-free resting state functional connectivity. Cool. So they basically talk about the various types of MRI that they're going to investigate for potential use here. Potential brain plasticity effects of BCI, right? We already talked about this. Yeah, cool. Cool, cool. Yeah, so they're basically just hypothesizing this like retroactively. Um, it's kind of cool when you see people like explicitly say like their hypothesis. I feel like in machine learning, there isn't much of that, just because of the nature. It's like the nature of machine learning research. It's more like engineering type than like scientific. Um, so you don't really see people saying like I hypothesize that X. So it's kind of nice seeing like these hypotheses, even if they probably were written after after the experiments were completed. Um, but yeah, yeah. Yeah, so they're basically just saying they expect the MI and ERP BCIs to impact uh, their respective regions, right? Ethical approval, blah blah blah. Okay, experimental design. So first they had the beforehand MRI, right? Then they have the EEG BCI stage where they actually stimulate for an hour, and then they have the post MRI. Everything is the same except for the motor imagery BCI group versus ERP BCI group. Um, and the MRI session includes both structural and functional scans. So in the MRI session, they have a T1 weighted anatomical scan followed by resting state and functional EPI scan. So I don't know what that means. I guess we might find out. They might not tell us. It might be like a really fundamental kind of thing in fMRI. I have no idea. But we will see. Okay, so the functional blocks during which they were actually stimulated with the BCI were each of 2 by 10 minutes. So I'm not sure if that means 20 minutes or they did like 10 minutes and then they had a break or something. Yeah, okay, imagine movement. So for the motor imagery one, they imagine movement of each of these like limbs. And then yeah, 20-second block design, right? So these are just like specifics. Here's like a diagram of how this works. So first they take an fMRI beforehand, like sorry, a ton of different fMRIs, right? They have I think this is the structural MRI, the anatomical one, and then the functional one is over here. And then they have the EGBCI session and then the same thing afterwards. Cool. More kind of specifics about their data acquisition. I think this is also specifics, like details. We don't need to go over this, I think, but we might come back here. It looks like there is some relevant statistical type of stuff over here that we might want to take a look at later on. Actually, like wait, when's the next session? Yeah, okay, let's just go through this and then we can pause for questions again. EPI data pre-processing. So they pre-process the data for the fMRI, right? Because fMRIs are like entire images with like tons of different traits. So there's like, I would assume you do need a lot of pre-processing to be able to deal with them correctly. Okay, and then here they talk about, I think, yeah, so this is what the plasticity depends on. But before we jump into this, yeah, like are there any questions about the setup here or anything up to this point? No questions. Okay, cool. So I think this is sort of an interesting, like important thing right here. Analyzing the functional connectivity between like these neural regions, right? Because that's how we determine whether the brain actually had like plastic changes or not. So they first use this eigenvector centrality mapping thing that characterizes each voxel's connectivity within the whole brain. So like, I'm not sure exactly what this is, but I would assume it somehow correlates a voxel, right? Which is like an individual, like part of the image that lights up, like they have some images down here of the fMRIs, right? Like each of these, yeah. So like you can see there's like a granularity of the fMRI, right? It's higher than a pixel, but it's like that's like a voxel, right? A single thing that lights up, a single unit that we can read off of an fMRI. That's what a voxel is here. Oh, and they mentioned this again, so I think yeah, this is going to be important. Where did this go? Over here, yeah. Right, so they compute a centrality map for each resting state block and a z-standard transform. Okay, this might just be a whole lot of jargon, and like this is not standard machine learning jargon. I think it is more like neuroscience stuff, so I'm not too sure what each of these specifically means. They use a paired t-test. That's like a statistical method. I know what that means, but the rest of this is indeed kind of cryptic. Maybe they'll talk about it later, but I think also these citations are going to talk a lot about these specific kind of methods that they're using. I guess the basic idea over here is just how do we determine what neurons or what sections, like groups of neurons, are firing in connection with other neurons. Yeah. Cool. They calculated spatial correlation maps with MATLAB. Yeah, so I think the goal here is just like, yeah, finding correlations between regions of the brain that fire together. So for each subject of both groups, they had a first level GLM. For each regressor, the imagination was well. Yeah, I think this is also like just specific details over here. I don't think we have to get bogged down too much in these. Yeah, and then this is, yeah, again. So I think this entire section was largely just details. But yeah, let's look over here in terms of the MI-BCI group. This is a bit interesting, I think. Right hand versus feet motor imagery. Different movements with each of the limbs. Interesting. So they were asked to choose the movement that caused more intense feelings on their body. I wonder what that was in for. Why did they ask that? Okay. Okay, so for the online classification task, every 40 milliseconds features were calculated as the log variance of the bandpass and spatially filtered data. So basically they're like chunking it into 40-second blocks, right? And then they are they're classifying it with just like an LDA classifier, which is like a type of LDA, linear discriminant analysis. There's also latent Dirichlet allocation, which is like a thing in NLP, but it also is spelled LDA. So yeah, that's the thing. But yeah, there's just some kind of ML classifier, right? It's like a classical ML thing. It's not a neural approach. Yeah, cool. All right, so basically, based on the results of this classifier, they show some feedback back to the user. So this is where the core feedback is happening. So we ask the user to like do some motor imagery, right? Then we feed the fMRI scan of that into this is EEG. Because they're doing like a bandpass and whatnot. I'm not sure why they never explicitly state the. Oh yeah, they talk about the EEG up here. Cool. So it's a 62 electrode setup with an EEG cap. All right, that makes more sense. So yeah, where were we? Over here. So they basically pass the EEG data to a classifier, and then based on the results of that classifier, they move the cross up or down on the field. So basically, the goal of the user is to like move the direction of the arrow in some certain direction. Yeah, so the user is presented an arrow, right? And they have to then do some motor imagery to cause this other cross, right? There's another like fixation cross, cause the cross to turn in a certain direction based on their motor imagery. So that's where the entire feedback kind of loop happens. Yeah. Two subjects reported trying to use eye movements, and they were eliminated from further analysis. Yeah, that's probably important, but also kind of brutal. So an increase in BCI. Okay, wait, so three different methodological adaptation levels. So they have like different levels of this. But then they're saying that in the group of users who found it difficult to achieve accuracy, right? They were saying like the 30% earlier, when they increase the difficulty or something, they found that there's an increase in BCI control among those users, which is kind of weird. Maybe I'm misinterpreting what they're saying here, but yeah. The first level oh yeah, so here they talk about the different levels, right? So there's a simple level where they're just calculating the logarithmic power values over certain like EEG bands. Mu and beta are like two EEG bands, right? There's like a ton of different EEG frequencies, right? That's like not a sine wave. Wait. Beta might look something like this, like mu might look something like this, right? They're just like different frequency bands. And then they are basically taking the logarithmic power values of these bands. So like if the beta is super powerful and the mu is super weak, right? The amplitude is super low, then they do something. So that's how this first one works. For the second level, okay, for the second level, they use CSP, common spatial patterns, which is it's technically machine learning, but it's very specific to neuroscience. Basically, it allows you to discretize a signal across spatial regions. So if like we have 62 electrodes we have like 62 electrodes across, we have 62 electrodes, right, across someone's brain. If we're seeing some signal over here that's like pretty high, and over here that's like also pretty high, but like the signal is like very weak over here, then we can use CSP and say, okay, the signal is probably strongest over here in this region of the brain. So they added this CSP filtering layer for their second level. And okay, level 3 consisted of logarithmic band power of fixed CSP filters with LDA adapted in an unsupervised manner. So it's some just like kind of improvement over that as well. So here they're using LDA with shrinkage of the covariance matrix. Yeah, so I mean, the basic idea is that they're just upping the predictive power of their classifier with each of these levels. Cool. Okay, so now here's the interesting thing. Let's see how yeah, just in terms of speed. Yeah, I think we're getting pretty close. We can skim some of these anyway. Cool. Okay. All right. So they had a spelling task for the ERP one. So we've just been reading about the motor imagery one so far, right? So for the, let's just skim this one. We don't need to go too depth in this one because they said this one had like higher classification accuracy and whatnot. But the basic idea, I think, is that they have a speller. The user has to check 1 out of 30 different symbols. Okay, interesting. Yeah, you guys can skim this right now if you want, but yeah, okay. Cool. Cool. It's basically just a speller where each of the images on, I'm surprised they don't have like a image for this, like the speller thing. That would be way better. Did you guys see anything over here? That sucks. Do they not have an appendix? No. Okay. Wait, did I see a figure? No, okay. All right. I mean, yeah, this basically just seems to be they have some kind of speller where the user has to pick a disk that they like a circle that has a specific symbol on it, and then the like feedback loop happens over here, right, for online and offline ERP classification. Wait. Oh yeah, over here. The user's focus on the target led to a notable increase in P300 amplitude that could be extracted from the EEG. So when they focused on a specific disk, right, that they wanted to put into the system, then like the P300 amplitude would increase in that specific region of their brain, right? Remember last week or maybe the week before we talked about how you can actually localize signals in the occipital cortex. You can tell where someone is looking based off of like where something is appearing in someone's field of view based off of activity in their occipital cortex. So I believe that's what they're doing here. But it might also just be like the other speller thing that Chris wanted to take a look at, right? Where you have different disks and they're fluctuating at different levels, like different frequencies, and then we can extract those from the brain based on just like ERPs. Okay, so yeah, that's the idea here. And then they have some more data analysis for the motor imagery stuff, right? Butterworth filter. We're using this as well for our project. Cool to see that. Cool. All right, now let's hop into results. Yeah, any questions about this? Yeah, sorry, a lot of this was just like meaningless details. Some of it was definitely important. Like I think it's a really important thing to understand, like the feedback loop, right? Where the person thinks something, right? And we read the signal, right? We put that through a classifier, and then we change something on the screen, right? So like that like shows up some weird symbol or something, and then they see this, and now they respond to that with their brain, and then their brain outputs something. Does this loop make sense? I think this is like one of the kind of core concepts in this, to be able to understand how the plasticity is actually induced. Okay. Oh, there's a question in chat. Okay, awesome. Okay, if there's no other questions before the results, let's hop into this. Okay. So first they're gonna look at the EEG experimental part, right? Like during the BCI when they're actually recording the signals, and then they'll talk about the results of the MRI sessions. Cool. So there's these 4 different runs for each of the groups, right? There's like the motor imagery group, right? And the ERP group. The horizontal bar represents the mean value for one run, so that the accuracy rate they have with the BCI feedback. You can see with the motor imagery, they get like consistently better over time. Whereas with the ERP, so now that, wait, okay, now that thing makes a lot more sense. Remember earlier we were like unsure of why they got better during the levels. It's basically because I think the ERP task is just so easy that by the first run, the user had already mastered it. Whereas with the motor imagery task, it takes a few turns for them to really get used to it, and then it like plateaus afterwards. Whereas with the ERP, it like just plateaus from the start. Okay. But yeah, you can see how the performance on the BCI task, like the performance itself isn't really what they care about here, but you can still see how it's pretty cool that they're getting something like 85% accuracy on these BCI tasks, which is pretty cool. Completely non-invasively again. Again, like EEG signals are really information dense for some things, like motor imagery or ERPs. They're really powerful, but then there's other things where like you can barely say anything based on the EEG at all. Like for language, actually there's this thing called N400, the N400 signal. It was another option for the paper reading this week, and it is sort of correlated with language, but generally you can't do like synthetic telepathy kind of stuff with EEG signals, whereas you would be able to do something cool with motor imagery. Cool. Okay. Let's look at the diagram first, and then we can read this. Okay, so over here, I think, what are they showing? Spectral analysis on the optimal channel of the left hemisphere. So number of selections per channel. Yeah, I don't know what that means. Yeah, you know, I don't read these beforehand. I read these on the fly. So I mean, it's sort of in the sense, like it's sort of good in the sense that you get an idea of how, like the process of reading a paper and what that's like. But then we also have things like this where I'm just like, I have no idea what that means. Number of selections per channel. It's such a simple phrase, but I'm not sure what it's referring to. Let's look over here. Yeah, this doesn't really say anything. I think by optimal band, they mean the mu band, right? Or the optimal. I guess they're just saying that they found that these channels over here had the most high correlation with the actual neurofeedback. I guess. And then here they have these activations for each of the motor imagery tasks. And yeah, you can see the frequency band, right? So in the mu band, I assume over here around 10 hertz, that's where we have this highest activation. Yeah, cool. Yeah, the motor imagery task here is like honestly harder to understand for me than the ERP one. I think it's just because I've read like a decent amount of ERP, like visually evoked potential stuff before, but not much motor imagery stuff. So I hope this makes a bit of sense to you guys. It just barely makes sense to me. Okay. MIBCI and ERP-BCI increase T1 weighted MR intensity. So this is like the main kind of finding, right? Let's look over here. So on the left, there's that motor imagery BCI group. On the right, there's the ERP BCI group. And then let's just take a look at the MI one beforehand. These are like different angles of the brain, right? Like from the back, from the side, from the top. So here, I think they're basically showing rest post 1 and rest pre 3. Oh, I think what they're showing is that what is rest post 1? I mean, so like this first one, I assume means that there are things that fire, like here they're showing the regions of the brain that fire at a higher rate afterwards than beforehand. Rest post 3, rest pre 1. Okay, well, all right. We need to find out what those mean. Rest post 1 minus rest post 3. Yeah, that totally makes sense. But what is rest post 3 versus rest post 1? Do you guys remember seeing this terminology up here? I remember they had that like timing thing. Rest post 3, rest pre 1, and rest post 1 and rest pre 3. Okay. So I think the post 1 pre 3 is basically right before and after, like adjacent to the EEG BCI session, and then the pre 1 post 3 is more like later on or like earlier before, just to see like if it persists after like 30 minutes longer. I think that's what they're trying to show here. Yeah, you can see how directly after the task, right, the firing is significantly higher, right? You have all of these different regions that are lighting up, whereas before they didn't. Whereas over here, after like another 30 minutes, it starts collapsing a bit. Like there's certainly changes in firing pattern, but it's not as significant as like right after the task. And it's a similar story for the ERP BCI group, except over here it's definitely retained a lot more, I would say. Like there's a lot of regions that continue to fire even afterwards, that didn't fire right after, which is kind of interesting. Maybe that has something to do with like how easy, like how the ERP task is easier. What do you guys think? Why does the ERP task maintain higher plasticity afterwards, whereas the motor imagery task seems to be like almost forgotten quickly? Any thoughts? Yeah, I think it probably just has something to do with how one is easier than the other. Also, motor imagery is a lot less symbolic, right? And when you have something that isn't really concretely connected to a simple symbol that you can just like call into your into your mind, I feel like things get a lot harder in terms of like remembering them. That's the thing about like words too, right? It's like almost words are sort of like bookmarks for ideas. And when you can't think about something linguistically or symbolically, you can't really maintain them. And I think that's sort of what happened with this motor imagery task, but maybe I'm just like extrapolating too much. I'm not sure. Oh, also, yeah, here they talk about like the regions, right? It's like spatially specific. Let's look at that again. Yeah, so you can see how, so where's the, so this is the occipital lobe, right, in the back of someone's brain. And you can see how there's significant changes in firing pattern there in the occipital, sorry, for the visual task over here in the back. Same story over here, right? There's just a different angle. And like different time thing, but yeah, same idea. Over here, not so much. Wait, actually, I'm not sure. Am I looking at this backwards? No, I think I'm looking at it right. Which side's the front and which side's the back? This side's the back. Yeah, this side's definitely the back. All right, so yeah, never mind. I was wrong about this. Wait, is the back? Yeah, honestly, I don't really look at these fMRI scans too much. Usually I just do like EEG stuff, but this is definitely an interesting kind of question. This does seem to be the front, because this is just like way too narrow to be the front of someone's head, I feel. So yeah, okay, I'm gonna say this one is like a bad kind of demonstration, but you can see how over here, right, like in the occipital lobe, there's some firing. Over here, definitely, and over here, definitely. Whereas over here, most of the activity is along the top of the brain, right, in the parietal lobe, and sensory motor cortex, which are like this band across the top of your head. And that's where we see most of the changes in activity over here. Cool. Here, I think they're just like talking about that stuff again. They're just doing between group comparison over here. I'm not sure that's too relevant to what we're interested in. Okay. Yeah. Yeah, I mean, here they're just saying that the BCI like explicitly only correlates with this specific measure of plasticity that they were using, like connectivity that they were using. It's like, it is pretty fair to extrapolate this to say BCI modulates like functional connectivity in the brain, but I think they're basically just explicitly stating that there could be other measurements of functional connectivity. And the one that they use is this eigenvector centrality mapping, which is like the only thing that you can say the brain explicitly, like the BCI explicitly changed, just because of the like the nature of science, I guess. Yeah. Cool. Yeah, I think they're just like repeating stuff over here, right? Right, it's like sensory motor for the MI group, but not for the ERP group, et cetera, et cetera, et cetera. Yeah, I mean, same idea over here as well. Bold, so there's something called a bold activation. Not sure what that is. Maybe it seems to be some kind of sensory motor correlated signal. So after the motor imagery BCI, there were what? Oh, so only for the right hand, when the MIBCI group was asked to use their right hand, there were increases in this bold activation, but for left hand and feet, there were no significant activations. Maybe this has something to do with your dominant hand. That would be kind of specific of a signal, but yeah, interesting. Cool. Okay, this is another kind of important thing I think over here. They talked about this SGNR squared value earlier. I'm not sure what that means, but they say the discriminability of the two tasks in the EEG. Yeah, I don't know what that means. What do you guys think? Does that make sense? The discriminability of the two tasks, like how you're able to tell apart, how are you able to, maybe it's like, are you able to tell based on the EEG whether someone is trying to do the what's it called? The motor imagery task or the ERP task? Maybe that's what that means. I'm not sure. Yeah, this is the same idea. Yeah, I don't know. This paper is honestly a lot more cryptic than the other ones we've gone through. This one's like definitely like hard neuroscience, whereas earlier we were doing more machine learning-esque kind of, or like BCI specific papers. This one is like, I think, sort of uses all the standard computational neuroscience tools and everything. So it's definitely a lot more hard for me to parse. Yeah, so this is the other thing that we're talking about in the abstract. I think this is also important. Precuneus and sensory motor cortex, two regions of the brain sort of correlated with motor imagery, I guess. And after this MIBCI, they show that there's increases in like correlated firing between them. Yeah, comparison to spatial correlation maps, yeah. So, whoops, but yeah. Yeah, in the MIBCI group, there's a lot more positive correlation, whereas in the ERP BCI group, not as much, right? It's like a negative correlation between those. Okay. Any questions before we just look over like the discussion of what this means, like future research and so on? Any questions about methods, results, everything? Yeah, if you weren't able to follow along with a lot of this, that's totally fine. I think the overall kind of idea of this paper was interesting, but I honestly, if I'm being honest, I don't think it was worth reading the specifics because it is like a very computational neuroscience oriented paper. And I don't think I gained much from it as like someone with not too much computational neuroscience specific, and especially fMRI. I think also a big part of it was just like I'm not familiar with fMRI as like a method that much compared to like EEG or EMG or something, which made it harder. But I mean, let's take a look at the discussion. It's fine if you weren't able to follow along with this. I think the overall kind of idea here is still definitely interesting, though. Also has some kind of like interesting philosophical implications. So let's see if they talk about that. Right. It just shows that in the visual group, they see increases in visual correlated areas. In the motor imagery group, they see it in sensory motor areas. same kind of deal. When we use the subject-specific features of run 4 and run 1, you observe similar correlation with T1 weighted MRI data and sensory motor cortex, but no correlation for ECM. So there's this, yeah, I'm not going to try to understand what exactly that means. Cameron, wait. The observed change in plasticity doesn't seem too different, but the change than the change when just learning any sort of task. Yeah, no, I think that's true. It's just that, I mean, I wouldn't know. I haven't looked at like fMRIs of how someone's brain changes before and after a like normal task, like learning how to like juggle or something. I haven't looked at fMRIs like before and after that. But yeah, I would assume that it would be a similar idea because, right, in this, it's the same thing, right? It's just here you're measuring it while they're being trained. Yeah, no, I think there is actually sort of a fundamental difference. In juggling, for example, your manner of interacting with the environment is going to be your hands, right? So you're learning how to move your hands, which obviously implicitly changes structures in your brain. But the manner in which you affect the environment is not like directly your brain. Whereas with the BCI in this like paper, it's like you're literally affecting you're learning to use your brain to affect the environment. And they're showing that even if you aren't learning to move your hands or anything like that, when you learn to use a BCI, it's also going to cause these changes in plasticity, which I think is sort of intuitive. You would expect that to happen, but yeah. Okay, let's just skim the rest of this. Pretty much done almost. Increased hippocampus side in London taxi drivers. That's interesting. Could maybe look at that paper. I don't know how they even decided to start thinking about that. Maybe because like the hippocampus is sometimes correlated with like spatial reasoning. So yeah. Cool. Just talking about like other research in this space, I guess. Actually, no, this is basically what Cameron was just talking about, right? Like when you're learning other tasks, it causes these structural changes in the brain that you can pick up with MRI. That's what they're talking about here. Yeah, so they kept talking about this MR signal, right? I think this they're talking about like the MR signal is connected somehow to structural changes in the brain, right? Like neuroplasticity. And they don't really understand why the MR signal gets increased when those plastic changes occur. Interesting. Microglial invasion. Microglia are like auxiliary brain cells that like assist with certain tasks and like cleanup and stuff. That's like the main way they're thought about. But they could also have a huge role in neuroplasticity. Neuroglial transmission has been shown to be fundamental in maintaining learning-induced cortical stability. Interesting. So they're saying that maybe these like these MR signal changes that happen with plasticity are due to like movement of the neuroglia, which cause this MR signal somehow. So they're basically saying that their findings could reflect this early change in early phase of brain plasticity, right? So like, because now they're able to measure the brain changes while the person is actually completing the task. So they can like pull out the MR signal and say something about that while they're actually learning about it. Cool. I mean, this is also kind of interesting, right? Yeah. Early events of brain plasticity, such as local accumulation of microglia. Nice, nice. Here, I think, so they just talk a lot more about the T1 weighted MR intensity, sort of all over this, and like the ECM stuff. So those are like the kind of the two key like metrics they use to quantify this neuroplasticity. Yeah. Yeah. Just like going ahead, like going through all the different research and showing how it connects to everything else and how they could explain the actual changes in neuroplasticity. Oh, okay. Interesting. I will be checking out those papers of my own interest, but that's an interesting kind of thing. I'm working on like syntax parsing, like cognitive models of syntax parsing research right now. So that's sort of relevant. Yeah. Cool. Yeah, I mean, now I think they're just repeating their final findings. Long-term neural reorganization. That's an important future research area. And how they could actually apply this for therapeutic uses. Right. So yeah, they close on this note about therapeutic approaches. Yeah, I think that's about it. Any questions about the discussion, right? Any of the kind of extrapolations they made based on other research or just on the overall paper, or thoughts or comments? Definitely, I think we're not going to, at least I won't be able to talk through MRI papers in the future. I think I definitely need to learn more about that before I try to go through research on it. So we'll probably be sticking to more, if we do like more computational neuroscience stuff, it is probably going to be more EEG based and whatnot. Yeah. Any other thoughts, guys? Any opinions on what you want to go over next week? Okay. Okay, cool. That's about it.